Okay, hi everyone. Thank you for all coming here for the last session, even though I had to like switch up the time. Something just came up last second. So thank you guys all for still coming. Okay, today we're gonna be covering microbiology and genetics. Even though it just says microbiology on the website, I decided that since we haven't really covered genetics yet, and it's like a pretty big part of biology, I just wanted to like throw part of it in this class. It should be really fun. So let's get started. Yeah, okay. So I think I always do this every single time, but this time before we begin, we actually have two pretty big announcements. So the first one is that our math contest winners are out. Oh my God, who is that person who keeps staring? But anyway, congratulations to all our contest winners. The top 9, oh, I think I forgot to put the 10th one, but the top 9 are here. And for the full results, you can click on, just go to the website, and then there's like a little drop-down menu where you can click on the math contest results, and then you can see everything. Okay, Daniel, this class is about biology, but we're on our 4th class, and our 4th class, we're gonna be talking about microbiology and genetics. Okay, anyway, congratulations to everyone who won. Next, let's move on to our second big announcement is that we are having 2 weeks of just special classes. They're not taught by the Flash Math staff. We actually recruited some other new teachers who are going to be teaching 2 weeks of special classes. So if it seems like SAT, math, writing, and world history are something you would be interested in, definitely attend those classes if you have time. Okay, so now let's move on to what we're actually going to be talking about today. So the first topic is microbiology. So first, we're going to talk about what are the different types of microorganisms. So microbiology is, in fact, the study of microorganisms and microbes and all that. So the first one, of course, is bacteria. Bacteria is probably the most universal microorganism that you've heard about. So here's some very important characteristics of bacteria. So first of all, they're prokaryotic. And for those of you guys who are not certain, like who are not familiar with the difference between prokaryotes and eukaryotes. So prokaryotic organisms are non-nucleated, meaning that their cells do not have a nucleus. Meanwhile, and they don't have organelles except for ribosomes. But on the other hand, eukaryotes, the cells are a lot more complicated. They have a lot of organelles and they have a nucleus. And the DNA and the genetic material is located inside the nucleus in eukaryotic cells, but in prokaryotic cells, they're floating around in the cytoplasm. Okay, so bacteria are prokaryotic and unicellular, meaning that one organism is just one cell. Most bacteria are actually beneficial. I think this is like a little bit of a common misconception that bacteria are super like pathogenic and they're harmful, but actually 99% of bacteria are helpful. They're found in your skin, in your intestines. Some bacteria are used by industries to help eat oil to get rid of the oil spills to help protect our environment. Some bacteria are used to convert nitrogen in the soil to help plants. So bacteria can be really helpful. But again, there's also some really pathogenic ones. One of them that is really pathogenic is Staphylococcus aureus. It causes all sorts of diseases from skin diseases to hospital infections to food poisoning. It causes a lot of things. So that's a very important bacteria. Okay, so here are some examples of bad bacteria, as in they cause diseases. So Streptococcus pyogenes causes strep throat, food poisoning caused by E. coli and Salmonella. And if you attended our public health class or my public health class, you probably remember that our entire outbreak investigation was based on an outbreak of food poisoning caused by E. coli. So that's that. Okay, Clostridium tetani is a very special disease in that it cannot be transmitted from one person to another. And when someone gets tetanus, then their muscles start having paralysis and can't move, and then their face is like stuck in a really weird, creepy position. So yeah. Okay, Bordetella pertussis is whooping cough. So whenever they cough, they sound like it sounds like a whoop. So yeah, that's the very hallmark of that disease. Next, we have Salmonella typhi that causes a very, very severe form of food poisoning in which you get fever, and the fever is really bad. So next, we have Streptococcus pneumoniae and Haemophilus influenzae. These two cause pneumonia, even though pneumonia is like a very broad category of lung diseases. These are just some bacteria that cause them. Okay, let me check Q&A real quick. When is the special session? So we had a special session on a few days ago on Scioli. I think it was 2 days ago. That was if you had 40 points or more. The special session today is at 3 o'clock, and it's if you have 50 points or more. So yeah. Okay, Daniel asked, what about a virus? We're getting into that real quick. Okay, next we're going to talk about archaea. Archaea, you may have heard of them being called as archaebacteria in the past, but recently it's been renamed as just archaea. They're also unicellular, meaning that one organism is just one cell. And the difference between archaea and bacteria is that archaea have a very rigid cell wall outside of them to protect them from extreme circumstances like high heat, high salinity, high pressure. So if you've been to like a geyser or a geyser, sorry, my bad, like in Yellowstone, there's a lot of archaebacteria underneath the hot springs. Okay, and archaebacteria or archaea are not pathogenic. So for example, methanogens in a cow's gut produces methane. So you may have probably heard about like how methane is really disgusting to the smell, and that's because the bacteria called methanogen are actually producing that. Okay, next we have viruses, which is what Daniel was talking about earlier. So here we have viruses. As you can see here, we have like some cell that's in purple, and then around it are like yellow orangish viruses. So viruses, unlike bacteria or any of the, well, most of the other microorganisms, they're non-living. There's actually been a lot of debate among scientists regarding whether or not viruses are living, and most have come to the consensus that they're non-living. The reason behind this is because they can't actually produce offspring on their own. Viruses are very simple. They're only consisted of a protein coat surrounding their genetic material, and the genetic material can be either RNA or DNA. So they're very simple, and they don't really have a means as to a way to reproduce. So the how they reproduce is here. So you can see here, the virus hijacks the cell. So the orange thing is the cell, and then the purple thing inside is the DNA. So then the virus hijacks the cell's DNA and integrates part of it, and then uses the cell's creation of protein machinery, so like their whole transcription translation material, to create copies of themselves. So they make the cell do all the work for them. And after the cell has created different copies of the virus, then the virus can proliferate and come out of the cell and create extra copies of itself. So that's why it's non-living, because they can't really do anything by themselves. They need like a crutch of another living cell to do so. Okay, someone asked about, what about viruses to make vaccines? Are they beneficial? Well, that is something that researchers are still trying to research in. So for example, for the COVID vaccine, the way they take vaccines is they would take the virus. So in this case, the SARS-CoV-2 virus, they would try to find like the membrane proteins that make it like special to the cell so that the cell can recognize it. After they get like those specific membrane proteins, they try to make a dead version of them. So or they may just kill the virus and make like a solution with the viral proteins so that whenever they inject a person with a vaccine, the person's immune system can see these viral protein. Sorry, wait, what just happened? My bad. Let me redo this real quick. But anyway, they can make another copy. And then wait, give me 1 second. I messed up something. Okay, now I should be able to share screen again. Okay, anyway, okay. So then the human immune system can recognize these viral proteins and start making these antibodies against them. So whenever they get infected with the real virus, they already have defense against the viral protein. Can you kill a virus if it's not alive? Technically, no. because it's not alive. So they would just deactivate the virus in this case. Okay, any other questions? I don't see any. All right, anyway. So you don't really need to know specifically exactly every single step of this cycle, but just know that the virus hijacks the cell's protein-making machinery to make copies of itself. Okay, next we're going to talk about a very special type of virus called a bacteriophage. Oh, it's kind of lagging, but I think it shows up now. So bacteriophages are very special in that they don't infect eukaryotic cells. They actually infect bacteria. And they can take parts of other, well, they can integrate their own DNA into the bacteria, causing bacteria to turn into these more virulent forms and possibly mutate. So that's just something special. Can you talk about Mottle's laws? I'm not sure what you mean by that. So yeah. Okay, so here are some examples of viruses. And as I was saying earlier, oh, Mendel's. Yeah, I'm talking about Mendel's laws later. Okay. Measles is an example. And just to note, as I was talking about bacteria earlier, I said that there's a lot of different helpful bacteria. But on the other hand, viruses, for the most part, are all harmful. So here are some examples like polio, COVID, varicella zoster causes chickenpox, and the common flu is caused by a lot of different types of viruses, but the three of the most common ones are rhinovirus, adenovirus, and coronavirus. And the last one is influenza virus causing the flu. Okay, fungi. So fungi are very special in that they are the only decomposers. Oh, Felix asked, isn't CRISPR made with the bacteriophage? Oh my God, I could go all day about CRISPR. I love it. What is going on with is somebody not letting me share my screen or something? Okay, so fungi, I'm gonna talk about the CRISPR thing first because you guys know about it. But basically, wait, why is my screen not sharing? Can you see my screen? Yeah. Okay. Now you can, right? So basically what scientists did was they found in bacteria that there's like these little immune system, there's like a mini immune system. Stop spamming, whoever that person is. I'm gonna kick you if you keep spamming. Whatever. So then the immune systems they found that inside the bacterial DNA, there are parts of virus DNA sprinkled inside. And that is basically what CRISPR is. It's like a immune system for bacteria. Okay, anyway, fungi are unicellular, although some are multicellular. Like mushrooms are multicellular, but baker's yeast is unicellular. They decompose, meaning that they take dead or decaying matter and they take the nutrients out of it and restore it back into the ecosystem. Okay, so helpful fungi include baker's yeast, which is used to have bread rise. Other uses of baker's yeast, there's a lot of them, but yeah, okay. Harmful fungi include stuff like Histoplasma capsulatum that causes a very rare disease called histoplasmosis. And Candida albicans causes thrush and yeast infections, if you may have heard of this. What does yeast decompose? Good question. So yeast actually undergoes a process called fermentation, in which they decompose sugars down into carbon dioxide, and all the carbon dioxide air accumulating inside the dough is what causes it to rise. Good question. Okay, next after, cool, I guess. If you know someone with athlete's foot, so athlete's foot is another fungal disease. It's caused by a fungi known as Tinea pedis. So Tinea causes a bunch of different like fungal diseases. There's one on the head, there's some between the fingers, and there's also one between the toes, which is athlete's foot, and it's like disgusting, but yeah. Next we have protists. Protists, some are unicellular, some are multicellular. Unicellular ones include, as you can see here, there's like that one on the very bottom left, that's Giardia. And if you go above it, there's like a little amoeba thing. But if you go to the very top and the very top left and the very top right, those are examples of multicellular protists. And seaweed is a very common example. Seaweed, you might think is a plant, but actually it's a protist, which is pretty cool. Okay, there's two types of protists, animal-like protists and plant-like protists. So an example of an animal-like protist would be a paramecium. There's not really a picture of that here, but euglenas, I think, are two. And then there are giardias, as you can see on the bottom. Plant-like protists are what I was talking about earlier, like seaweed and algae. Okay, specific examples are Plasmodium falciparum, Plasmodium. That is the disease that causes malaria. And Plasmodium is a very special protist in that it has a very complex life cycle. It has parts of its cycle inside the human. Parts of it is very cool if you want to look into Plasmodium's life cycle. It's pretty complicated. Oh, someone asked, should I go to the special sessions at 1 or stay for biology? I'm not sure what time zone you're in, but the special sessions end after biology, so just letting you know. I mean, sorry, the special session starts after biology. Someone asked, what viruses infect viruses? I'm not really sure of any certain viruses that infect other viruses as of now, because there's really no point in a virus infecting another virus, because the whole thing, like the whole reason why a virus would want to infect something would be so that they can make more copies of itself. But if it infects another virus, that other virus can't exactly make more copies of the virus, so there's not really a point in doing that. Let's see, are there any, I think I saw a question in the chat earlier, let me look. Does anything cause hysteria with the mind? Oh, good question. Well, the thing is, hysteria isn't exactly like a very well-defined disease. Hysteria is like it's like a lot of people freaking out, if it makes sense. So a historical example would be like the Salem witch trials, but I'm not sure exactly what you mean by hysteria, so yeah. Wait, what is the dude who keeps what's his real name? Because I can't really find him in the attendees. And also, whatever, I'm just gonna move on. Okay, anyway, parasitic worms. So parasitic worms are the next thing we're gonna talk about. These are actually macroparasites, meaning that you can see them with your own eyes. And so here on the right, we have a picture of a roundworm, I believe. Correct me if I'm wrong, but let me see if I can find. Okay, guys, I banned the guy who keeps spamming, so hopefully we're a little bit more quiet. Yeah. Oh wait, I did not know about virophages. That's a cool thing. I guess everyone has something to learn. Anyway, so next we have macroparasites, in which, I'm just gonna close the chat. Okay, if you have any questions, always just ask in Q&A if you have any. Okay. So then we have macroparasites. Most of them nourish off of a person's intestines, and then they can migrate off to their bloodstream. Why does this keep happening? Okay, next we're going to talk about prions, and prions are something you probably have not really heard about before, but they're very different, as in they cannot, they're not alive, similar to viruses, but they're even more simple than viruses. Prions are literally just proteins that are folded weirdly. So yeah, they cause other proteins next to them to also fold up weirdly. And some notable diseases are scrapie and Peru and mad cow disease, which is caused by when cows eat other cows' brains, which is disgusting, because some farm people put the brains inside people's feed, so that's not very good. And then sometimes it can mutate into a human variant, which is Creutzfeldt-Jakob disease, so that's pretty bad too. Okay, let's see. Someone asked, do parasitic worms come in undercooked meat? Sometimes. There's different types of parasitic worms. For example, one of them is called ascariasis, and that one does come from undercooked meat. And that is pretty bad because once you eat it, it can migrate off to your brain and cause your brain to like get messed up in simple terms. So that's something. Okay, note, many microorganisms are helpful. All have helpful species except viruses and prions. So prions, they always destruct the other proteins in your brain, and viruses are always bad too. But other ones like protists, we have things like seaweed. Fungi, we have things like mushrooms that are good to eat. And then we have, for bacteria, we have like E. coli in our gut to help us with, oh, someone asked I'll just go back. And then bacteria, we have the ones in our gut to help us with digestion. Fungi, we also have yeast, as Eric is saying. And then, what was the other one again? Was there another one? Archaebacteria, I guess. Archaebacteria isn't really pathogenic, but yeah. Oh, by the way, parasitic worms are also really bad too, because they literally have the word parasite in their names. So, yeah. Okay, so next topic we're going to talk about today is genetics. Genetics is a very, very big part of biology. Almost like wait, I feel like someone is hacking this Zoom. This never happens. Wait, I've literally been doing this for like a long time, and this has never happened before. So give me like 2 seconds. Yeah, I'm gonna call Rich real quick. Or it might just be my really bad connection. Hold on. Somehow this account stops being okay. I'm just gonna fix this real quick. Okay, now it works again. Hopefully. All right, anyway, I'm just gonna wait till this thing actually comes back up. So anyway, let's talk about genetics. Our genetic code is, for the most part, DNA. DNA is, you probably have heard teachers talking about how DNA is the blueprint of our body. And in some ways it is, because it stores all the genetic information. It encodes all your genes. It encodes protein. Then the reason why we say that it's the blueprint is because these genes specify the creation of proteins, and each person has a unique set of their own proteins. So for example, you may have a protein that creates like pigment for your eyes. And so that's how you have your own eye color. And that protein is, what the? You know what? I'm gonna start sharing from this account. So give me a second. I don't know why this keeps happening, but I'm gonna share from here. Sorry for doing this thing. Thanks y'all for being patient. Here. Okay, hopefully this is good now. Y'all can see that, right? Okay, the bad thing is I can't really see the chat anymore. So I'm gonna just, y'all, if y'all have any questions, just ask in Q&A, okay? Okay, anyway, so next we have translation, or sorry, wrong slide, my bad. So our genetic code is here, so this is where we left off on. Anyway, so here you can see a picture of DNA. DNA is double-stranded, as you can see here. It has 3 main components. What is going on? Okay, no, nothing what happened. Okay, so then we have, it's located in the nucleus of eukaryotes, and the segments are, the different strands of the DNA are anti-parallel, meaning that they run in opposite directions. So the way to characterize direction in DNA is to say 5' to 3' or 3' to 5'. The 3' and the 5' characterize positions of the sugar. And so one side would be going from 5' to 3', and the other side would be going from 3' to 5'. So yeah. I'm gonna make it full screen so you guys can see it better. Okay, so there are three main components of the nucleotides. So many repeating components called nucleotides, but we'll go on to the three main components in the next slide. So nucleotides. Nucleotides are the repeating parts of the DNA. As you can see in here, each one of these little bars are nucleotides. So here I have a diagram of a nucleotide. Over on the very right is the the nitrogenous base. And the nitrogenous base can be any one of four compounds: adenine, thymine, guanine, and cytosine. So after that, we have the middle thing in brown, which is the pentose sugar, and in deoxyribose. You can see that this H right here actually does not have an O next to it. And that's why it's called deoxy. Deoxy meaning that it took away an oxygen. So normally ribose has another O over here, but in this case it doesn't, which is why it is called DNA and instead of RNA. Okay, and then on the very left we have a phosphate group, and this is just another structural part of the DNA. So next, let's look back over here. You probably already know this, but adenine and thymine pair together. They hydrogen bond together. Cytosine and guanine hydrogen bond together, and this is how the DNA works. So you can see here, adenine and thymine are forming 2 hydrogen bonds. Guanine and cytosine are forming 3 hydrogen bonds. And if you don't know what hydrogen bonds are, I suggest you go back to my first class on biology where I talked about biochemistry. I explained hydrogen bonding in a lot of detail in that class. Okay, next thing that we need to talk about is purines versus pyrimidines. So purines have 2 rings. As you can see here, this is adenine. Adenine has 2 rings. Guanine also has 2 rings, so this G right here. And then pyrimidines instead only have 1 ring. So cytosine and thymine only have 1 ring. However, you need to have 1 purine and 1 pyrimidine to form a hydrogen bond in order for the inside of the DNA to be the same length. Because if you have 2 purines hydrogen bonding together, then that's too thick or too fat. But if you have 2 pyrimidines and 2 pyrimidines bonding together, that's too skinny. So you need to have 1 purine and 1 pyrimidine, and it just so happens that A and T hydrogen bond perfectly together, and C and G also hydrogen bond perfectly together. Okay, moving on. Oh, so here I just included some diagrams of DNA. You can see the hydrogen bonds occurring between adenine and thymine over here, and you can also see the hydrogen bonds between guanine and cytosine. So notice that there's 3 hydrogen bonds between guanine and cytosine, but there are only 2 between adenine and thymine. So what does that suggest? So in the chat, wait, am I even able to access the chat right now? So in the chat, even as I'm trying to figure it out, try to let me know what this suggests about the strength of the bonds between the different types. Oh, okay, I can see the chat on my iPad. So which one do you guys think? So tell me which one is stronger and which one is weaker. Okay, I'm seeing a lot of good answers here. C and G is stronger would be the correct answer because each hydrogen bond contributes some form of strength. So if you have 3, it'll be stronger than if you have 2. So hopefully that makes a little bit more sense. Okay, and on the bottom here, you can see here are the different structures of the purines and the pyrimidines. On the left, I just wanted to show you guys something that you probably do not know, but because I know when I was learning about this, I actually do not know about it. But the thing is, you guys have probably all heard about ATP, right? Type in the chat what ATP is. Yes, everyone is saying energy, which is correct. So energy stands for, or ATP stands for adenosine triphosphate. So the adenosine actually means like adenine almost. So the structure of adenosine triphosphate, which is ATP, would be an adenine over here and then 3 phosphates over here, which I thought was so cool because look at this bottom part that shows it. So just this thing right here. So just the base is called the nucleoside, and then adding 1 phosphate turns it into the nucleoside monophosphate, and then adding 2 turns it into the nucleoside diphosphate, and adding 3 turns it into the nucleoside triphosphate. So if this nucleoside right here was adenine, adding 1 would be AMP, so adenosine monophosphate. Adding 2 phosphate groups would be adenosine diphosphate, so ADP. And adding 1 more phosphate group would be ATP, so adenosine triphosphate. And every time one phosphate group is taken off, so every time it turns in from ATP to ADP, then energy is released. So that's basically how we get our energy, through hydrolyzing these different phosphate groups off of the different nitrogenous bases. Okay, Ishas, does it matter if the nucleoside is purine or pyrimidine? It does not matter at all. But ATP is just a more common type of nucleotide, or nucleoside triphosphate. There's actually a very common one called GTP as well, guanosine triphosphate. And guanosine is actually a purine as well. It's used in translation, so which we'll get to that later. Next, let's move on to what is RNA. So RNA, the most striking difference between DNA and RNA is that RNA is single-stranded, as you can see here. And also, RNA is not deoxygenated. So if you go back to this little diagram over here, you can see that if here, you guys can see my mouse, right? Let me know if you can see my mouse. Okay, good. So you good? So where my mouse is pointing, you can see the OH, right? With the OH, it would be called ribose. But without an O, that would just be called deoxyribose. So in DNA, there's no O, but in RNA, there is the O. So that's another important thing, not deoxygenated. Instead, it's just oxygenated. Single-stranded, of course. And another thing is that RNA can come outside of the nucleus in eukaryotic cells. Meanwhile, DNA only stays in the nucleus, and it's just meant for storage of genetic information. RNA is also involved in transcription and translation. Meanwhile, DNA is only involved in transcription because translation occurs outside of the nucleus and DNA cannot come outside of the nucleus. Okay, there are 3 main types of RNA that you should know. The first one is the one that we're probably the most familiar with, which is messenger RNA. That's the one that's shown on the right. That the messenger RNA is transcribed from DNA, and then it comes out of the nucleus, and it serves as a template for protein creation. Okay, next we have ribosomal RNA that creates ribosomes. So ribosomes are where we do our translation. And they are made of 2 main things, ribosomal RNA, or rRNA, and some other proteins. So those are the 2 things that make up the ribosome. Okay, Felix is asking me, what is the difference between RNA and DNA? Don't worry, we'll get right to that in the next slide. And finally, we have transfer RNA, which is tRNA, and that is also involved in translation. Okay, another important thing about RNA that you cannot forget is that instead of having thymine in RNA, we have a different nitrogenous base known as uracil. So look over here on the right, you can see that we still have adenine, guanine, cytosine. Those are all normal, but instead of thymine, we have uracil. So yeah. Let's talk about the comparisons between DNA and RNA next. So first, DNA double-stranded, RNA single-stranded. DNA has thymine, RNA has uracil. DNA stays in the nucleus, and RNA can travel outside of the nucleus. DNA is involved in DNA replication and transcription. Meanwhile, RNA is involved in transcription and translation. DNA has a deoxyribose sugar backbone, meaning that it has has an O. Well, no, it doesn't have an O in that 2' place for the 5' sugar. And then the RNA does have an extra oxygen there. So that's why it's not deoxy, so it's just ribose. Is it possible to create a living thing / make a human? Wait, okay. I don't fully understand what you mean. as in like they you can definitely create a human with like parents. I don't know. Okay, Sherry is asking, can viruses affect your DNA? Well, actually they can, because that's a very important thing, because the whole way that viruses infect cells is by inserting their DNA into your DNA so that your transcription and translation machinery will do all the transcribing and translating for the virus and create more copies of it. Okay, next let's move on. Oh, one more thing before I move on. I just wanted to say that, you know how DNA has thymine and RNA has uracil? Well, whenever DNA is getting replicated, the A matches with the T. But in RNA, the A will match with the U. Oh my God, David, I see your question. No need to spam it to me. Is RNA more likely to mutate? I'm actually not sure about this. Oh, actually, it depends, because sometimes DNA can get mutated by things called mutagens, like UV radiation. Meanwhile, RNA doesn't exactly subject to that, but correct me if I'm, you can probably Google that. It's probably more, you'll probably see more information on that. Okay, what happens to your DNA when the virus does its replication? Okay, well, your DNA, okay, the virus, okay, let me just rephrase real quick. The virus doesn't change your own DNA. It has its own set of DNA, and then your transcription machinery will do the thing for that virus, if that makes more sense. Okay, let's move on. Oh, let me check the Q&A. I think I forgot to check that. Can you go over dehydration synthesis? Good question about that. The thing is, I talked about that in my very first class, which is week 1. So check the video out on that. Okay, Isha asks, does your cell still transcribe your DNA as well? Yes, your cell always transcribes your own DNA. I think that's about it for the questions. So here is DNA replication. So give me a guess as to when do you think DNA replication occurs? So type it in the chat. Okay, I got a good answer. Continue typing in. Good. Okay, perfect. Everyone is talking about cell division, which would be the perfect answer, because that is when you would have to replicate your DNA. When one cell turns into 2 cells, you can't just have one copy of DNA. You need to have 2. So that's why you do replication during DNA or during mitosis/meiosis. Okay. Here are the different mechanisms involved in DNA replication. So notice that you're not creating an entirely new strand of DNA. You're just turning your old one into 2 by adding new stuff on each strand. Okay. So let's look at what's going on here. So first, let's look at this green thing on the very right. This thing is called topoisomerase. So notice that everything beyond this is like is twisted, and that's how DNA is. But this topoisomerase gets rid of the coiling of the DNA so that DNA synthesis can actually occur without getting all coiled up. Okay, now let's move on. We have this blue triangle, and this enzyme is called helicase. It unzips the DNA so that there's 2 different strands so that it can go. Next, we have these purple things. These are called single-stranded binding proteins. The purpose for these is so that it can hold the DNA back. It can pull it back so that the 2 strands that the helicase just unzipped don't go back together, and that synthesis can actually occur. Felix is asking, can something go wrong during replication? Yes, and I'll go over that right in a second. DNA primase. So this is very important, because one thing to know is that DNA replication can't occur without something called a primer. So DNA primase actually forms the primer, and the primer is just a molecule of RNA. So see here, this RNA actually allows the DNA polymerase, to this orange thing, to actually start doing its synthesis. So without this RNA piece right here, DNA replication cannot occur. Okay, moving on, let's look at this bottom strand right here. So notice that the DNA of the original strand is going this way. So it's going left, but the DNA polymerase is going right. And this relates to the whole anti-parallel thing I was talking about earlier. The two strands run in opposite directions. So then this RNA polymerase or DNA polymerase can just keep on going straight through, and there's no interruptions. And that is the reason why this is called a leading strand. Okay, but if we go up here, notice that this original DNA template is running from left to right. So then this DNA polymerase has to synthesize from, this top part is going from right to left. So then this RNA polymerase has to synthesize from, wait a second. Oh yeah. So it has to synthesize from left to right. So this has to go from right to left. Okay, sorry about that whole thing. But basically it's going in the opposite direction, but now it has to create these little Okazaki fragments because it can't just be a continuous synthesis of DNA. And so after each of these fragments is created, they're called an Okazaki fragment. And once a bunch of Okazaki fragments are created, this blue enzyme here is called DNA ligase. And this one is in charge of combining all those little fragments together into one continuous strand of DNA. So that's how DNA replication occurs. Any questions? Oh, Felix asks, why do the two strands have to not be separated at the same time? Why leading and lagging and not just same start? Okay, so that's a good question, because if you look at the DNA right here, you see this is 5' and this is 3', and the DNA is running from 5' to 3'. So look, let's just follow my mouse right here. This one starts at 5 and it's going towards 3, so in this direction. But remember how DNA is anti-parallel, meaning that if one goes from 5' to 3', the other must go from 3' to 5'. And DNA synthesis always occurs from 5' to 3'. So this strand right here can literally just synthesize continuously because it's always synthesizing from 5' to 3'. Okay, hopefully that makes sense. But now let's look at this top part. This top DNA strand is going from this top strand, the 5' is over here. See, look, this top DNA strand goes from this way to this way. You see it? But notice that they have to be going in different directions. So this bottom new strand has to be going from right to left in this bottom thing here. So, okay, but this DNA polymerase can only go in that direction. So like totally this DNA ligase, okay, this is kind of hard to explain, but this DNA polymerase first synthesizes a little bit over there, and then it goes forward and then starts from here and then synthesizes this little piece over here. And then it goes forward and starts synthesizing a little piece over there. That way it continues to synthesize in the same direction, but it still is synthesizing from 5' to 3', if that makes sense. So yeah, I'm not sure if that makes sense completely. So if there are still questions, let me know. Okay, David is asking, is Okazaki the guy who discovered it? I mean, probably. I mean, I'm pretty sure. Okay, correct me if I'm wrong though. Let me check Q&A real quick. DNA polymerase. So DNA polymerase is basically the enzyme that adds new nucleotides. It synthesizes new DNA, basically. Let's move on to transcription. So transcription, everybody type in the chat what they think transcription does. Or when do you think transcription is needed? Okay, Isha says like converting. That's a good answer. Let's see, any others? Yeah, basically that's correct. So whenever your DNA needs to actually be converted into RNA, that's when transcription occurs. Okay, let me check here. Why are some pieces falling off? What do you mean by that? Oh, I see what you mean. Transcription factors, I'll talk about that right now. So the thing is, transcription is very different from DNA synthesis because you're only creating RNA, you're not creating a new strand of DNA. So, so in eukaryotes, transcription must occur. Like the RNA polymerase is a lot cooler in prokaryotes because there's this sequence in the DNA called a promoter. and the promoter is usually like TATAAAA or something, and that's called the TATA box. So once this, in prokaryotes by the way, once the RNA polymerase finds this TATA box, then it can just immediately start making RNA. But on the other hand, the eukaryotic RNA polymerase is not as cool as the DNA RNA, the prokaryotic RNA polymerase. In eukaryotes, we have to have these things called transcription factors that bind to and next to the TATA box in order for a transcription to occur. So after these little blue, purple thingies bind to the place where the promoter is going, then the RNA polymerase can find these transcriptions factors and bind to them, and that's how it recognizes where to start. So that's why some of it looks like it's falling, but it's really not. It's just binding to the correct place on the DNA. Okay, let's move on to the actual part of transcription. So on the right, on the left is replication, which we talked about earlier. So let's focus our attention to the right. So notice that this purple strand or pink strand right here is the RNA. And this might be a little bit of a tricky question, but let me know if you think this transcription occurring right here is in prokaryotes or in eukaryotes. So like in bacteria or like in animal cells or plant cells. Just what is your guess? And also let me know why you think that. So I'm seeing a good mix between prokaryotes and eukaryotes. So type it in the chat why you think that. It could be, it doesn't really need to be super like cool of a. Okay, some say because ribosomes, but the thing is both prokaryotic cells and eukaryotic cells have ribosomes. But that's not really something. Wow, Dave, it looks complicated. Okay, that sounds cool, I guess. Yes, Isha is correct. So look, there's no nucleus here. And remember that the DNA must be in the nucleus in eukaryotes, which is why it must be a prokaryote going on here. So see these little red things that kind of look like strawberries? Well, they're not strawberries for sure, but these are ribosomes. These are places where all the mRNA is getting turned into proteins. So this is another special thing that you need to know about the difference between prokaryotic and eukaryotic transcription/translation is that in prokaryotic transcription translation, the mRNA doesn't need to leave the nucleus because there is no nucleus. So after the mRNA is made, the translation with the ribosomes can immediately occur. So they occur simultaneously in prokaryotic cells. But in eukaryotic cells, the mRNA needs to completely finish and then come out of the nucleus, then find the ribosomes, then start with the translation. So that is the difference between translation/transcription and eukaryotes and prokaryotes, which is also why this is prokaryotic cell. Okay, any questions about this? Let me check Q&A. No questions. Any questions? Nope, okay, cool, moving on. So now let's talk about eukaryotes since we were talking about prokaryotes earlier. So look at this little piece on the right. Notice that there are little dark red areas and orange/tannish areas. So after the DNA is transcribed into RNA in eukaryotes, it's a very like premature mRNA. And that's why it's called pre-mRNA. But this pre-mRNA needs to go through some fixing in order for it to be mature RNA or mat RNA. So first thing that it does is it adds this little green thing on the left, which is called the 5' methylguanosine cap, as I said over here. And then after it does that, it adds this black part over here called the polya-tail. And the polya-tail is literally just like hundreds of adenines, like one after the other. So A. So do you guys want to give an idea as to why this would occur? Like why they would want to add a cap and this whole long A sequence? Oh, okay. Someone asks, why does it seem like eukaryotes are super I think you mean inefficient, maybe, or in infection, but I'm going to answer the inefficient. So That's because eukaryotes are very like, they underwent a lot of evolution. So after one stage of evolution adds another piece of machinery to help prevent it from doing like weird stuff, to put it simply. So there's just been a lot of evolution with eukaryotes, which is why. So anyway, anyone want to, okay, David, I see you. I see your answer. No need to spam it. Any other people want to guess as to why we add a cap and a poly A tail to the pre-mRNA? Okay, good. I'm seeing some good answers. Oh, wait. Okay. Everyone is, well, almost everyone is saying that, okay, some people are saying something different, but the reason why is because we don't want like random enzymes in the cell to be degrading parts of the mRNA, because all the mRNA is important, like all the ones that stay are important in trying to make proteins, but there are enzymes that can actually harm the mRNA, which is why we need like a protection on each side. Okay, oh, also this black part, I think it's just supposed to be this part right here, but I think they accidentally like took out the poly A tail in the second part, but just remember it's still there, the poly A tail. Okay, and then these orange things are called introns. Oh wait, my bad. The first thing is they take out this part right here. And then they add the poly A tail, which is the red part, over here. So my bad. Okay, but then they take out these little orange tan things, and these are called introns because they don't code for anything important, and they're just there. So they take them out. The spliceosome is a special thing that takes them out. And then after that, we're left with our final product, which is the mature mRNA. So any questions about this entire whole process of RNA processing? I'm going to move on then. So next we're going to talk about translation, something I've been talking about for a long time. Yes, the final mRNA is just exons. Okay, so next we have translation, and this is the process of turning the mRNA into proteins. So this little diagram over here is a pretty good diagram. So each one of these little blue things is a molecule of tRNA. And on the very top, we have the amino acid that it carries. So each one of these tRNAs will recognize 3 nucleotides in the mRNA, and then it'll match to it. And each 3 mRNA specifies a specific amino acid. And then it just adds that new amino acid to this long chain that's already occurring. So that's how translation occurs and how the mRNA turned into protein. So yeah. So each triplet of 3 nucleic acids is called a codon, and each codon specifies a specific amino acid. And more than one codon can specify the same amino acid, because if you think about it, 4 to the power of 3 is 64, but there's only 20 amino acids. Okay, moving on. Mendelian genetics. So as someone was talking about earlier, they wanted me to talk about genetics, so we might have to hurry up because we have around 5 minutes left. So let me try to explain this as quickly as I can. So genes are expressed as a sequence of DNA on a chromosome. You inherit a chromosome from each of your parents. Oh, sorry about the lack of an S there on parents, but yeah. So there's different like types of a gene. There's a type that's dominant, and there's a type that's recessive. And each of these different types is called an allele. So what is the difference between dominant and recessive alleles? So everyone has 2 copies of a gene. If one of them is dominant and the other is recessive, then it'll just be dominant. Like the characteristic that the person will display would be dominant. Meanwhile, if you have 2 dominants, of course, it'll just be dominant. If you have 2 recessive, though, then you will display the recessive trait. So yeah, dominant masks a recessive. So let's look at an example. Rolling your tongue is dominant, so it's shown as R. So we typically write the dominant allele as the capital letter. And recessive traits are the ones that are written as lowercase letters. So being unable to roll your tongue would be a recessive trait, so we're writing it as a lowercase r. So let's say the dad's genotype, or the dad's like 2 different alleles that he inherited, were RR, meaning that both of them were dominant. And the mother has the genotype of 1 dominant and 1 recessive. So let me know in the chat, can the dad roll his tongue and can the mom roll his tongue? So let me know in the chat. Okay, good. I'm seeing good answers. Both of the parents can roll their tongue. The reason is because both, they have at least one copy of the dominant allele. So the question is, how many children can roll their tongue and what is their genotypes? So let's draw a Punnett square. So here we have the dad's genotypes on the top and we have the mom's genotypes on the side. So we just cross each one of them. So R, big R, big R, big R, big R, big R, small r, and big R, small r. So notice that every single child has at least one dominant allele, which means that all of them can roll their tongue. However, half of them will be homozygous, which meaning that both alleles are the same, and half of them will be heterozygous, meaning that they have one dominant and one recessive. So one thing to remember about homozygous versus heterozygous is that homozygous just refers to their alleles are the same. So even small r in this case would be homozygous. Meanwhile, big R, small r would be the only one that's heterozygous, if that makes sense. Okay, perfect. So I think we have, okay, before we move on to the end, I just wanted to say that if you want more practice with anything that I talked about today, it's all going to be in the homework. It's all going to be on the website. So yeah, okay. Okay, so for those of you guys who came today, I am so thankful for you guys. Thank you all for coming to all of our classes or any of our classes throughout this summer camp. And we just wanted to thank you guys so much. And also we want to help improve for our next possible event that we're gonna host. For example, like over the school year, we might do some classes. So we just wanna know you guys' evaluation and your, and just what you guys thought about our class overall. So you can just go to this QR code, scan this QR code on your camera, or you can just click on this link. It'll take you to the camp evaluation survey part. So I'm gonna stop my sharing so that I can actually see the chat because for some reason I can't see the chat right now. Yeah, but right now all I need is for you guys to do Q&A. Yeah, I'll send the link in the chat right now. Give me 1 second. Okay, no one spam right now, okay, because I'm sending the link in the chat. And if you can't find it, then you can also just go to the Kid Teach Kid website. There's like a little drop-down menu where you can find, under classes, you can find all of this stuff. Okay, anyway, any questions about anything I talked about today? I know it's a lot. Oh, yeah, sure, y'all can chat now if you want. Okay. Anyway, we would really appreciate it if you can attend the tests or the new classes that we're hosting in the next few weeks. Those are by some of our amazing teachers. We're getting some new teachers and they're going to be teaching some more humanities focused classes since most of our classes are like STEM related. So those of you guys who are also interested in like reading and writing / history, y'all can also attend those classes.